I'd like to thank the organizers for this opportunity. I've actually never been to the main RECOM event, and it's been really wonderful to see old colleagues and make new friends. I'm also really excited to be here representing population genetics. So traditionally, my group has gone to RECOM genetics. So I'm looking forward to telling you about some of the problems that we think about and hearing feedback, especially on the second half of the talk, about ideas we should be using that some of you may know about that we could benefit from. So as a population geneticist, specifically working on human genetic variation, the question that's at the center of my research program is this one, which is how is human genetic variation shaped by population histories? If we think about our own species, we're really shaped by a very interesting and fairly recent in evolutionary time shared history, during which our species emerged in Africa about 200,000 years ago, grew rapidly, and then experienced large-scale migration events, as well as secondary contact during more historical events, including colonization and slavery. So this shared history and the ability to tell stories about it using the genome and infer deep history using the genome is what led me to human population genetics. My group has developed a series of methods leveraging Gaussian processes and the sequentially Markovian coalescent in order to finally infer population size changes over time. More recently, we've studied things like the fact that the X chromosome is an introgression desert and leveraged sex-biased demographic processes along with selection and the inheritance of the X to help explain why this might be. So why is it that there's less archaic ancestry on the X chromosome, which others in the room, like Sri Raman, have worked on. We also use human population histories as a way to condition on neutral events in order to identify beneficial mutations. So if we imagine that this history has shaped our genomic background, then we might, when we're thinking about adaptive mutations that allowed our ancestors to deal with new diets, new pathogens, and new environments, we might imagine there's some picture like this one on the right now, where there are certain mutations that enabled this process. But in order to look for this pattern, we need to understand that our neutral histories will strongly influence the fate of new mutations and actually confound traditional tests for selection. And some of this work is also influenced by work from other people who are here, for example, from Adam Siepel's group. And recently, more and more methods, including our own, are leveraging the ancestral recombination graph in order to answer this question. But more notably, I think it's really interesting to think about our species as one that, in the traditional evolutionary sense, is very much not in equilibrium. And we know that all of these evolutionary processes leave their footprint, particularly in one important signature in human genomes, which is that of linkage disequilibrium, which we can think of as just Pearson's correlation. So just to show you what this sort of looks like, serial bottlenecks, which have pervaded our human evolutionary history and pervade the history of many organisms that we interact with, for example, domestic species and bacteria like H. pylori that have accompanied us on the diaspora out of Africa, predict an increase in linkage disequilibrium with genetic differentiation from Africa. So here's a picture of what this looks like from Noah Rosenberg's group in one of the first analyses of SNP-CHIP data applied to population genetic data sets. This is from the Human Genome Diversity Panel. So this is a set of 52 populations, individuals who are two-generation indigenous to those populations as an assay of human genetic diversity. And part of why I'm showing this now relatively old picture is that this pattern has continued to be robust as samples have increased and as marker types that are being used have changed. So what's happening here is on the y-axis, we have genotype-based linkage disequilibrium. So basically, you can think about this as correlations in genotype space in 10 kilobase windows. And you can see a very systematic increase with genetic differentiation from Africa. Here we're using geographic distance as a proxy for that in these putatively indigenous groups, that, as we can call them that. So what this means is that there are shorter shared sequences in African genetic ancestry individuals than in, say, indigenous American individuals, because recombination has had more time in African genetic lineages to break up those associations. But this fundamental systematic difference pervades all of us in this room, for example. And so what this tells me is that LD, which lies at the heart of the project in some sense of human genetics, which is identifying adaptive and disease-causing mutations, is going to differ systematically by ancestral background. So how do we incorporate this as we move forward in our methods? So I think for everyone who's here, but maybe particularly for the researchers from the US, it's really interesting to note that the National Human Genome Research Institute puts out its strategic priorities, has done every decade since the publication of the draft sequence of the Human Genome Project. And the most recent paper is a really interesting read, and I'd like to use that as a way to sort of outline what I'm going to try to do today. So in this note, there is a box called Bold Predictions for Human Genomics by 2030. And what it notes is that many of the most impressive achievements in genomics, when we look back over the last decade especially, now 2 decades out from the draft sequence publication, could hardly have been imagined a decade ago. And so now there are 10 new bold predictions. I also think it's important to note that the way this is phrased in the paper is that these are aspirational. These might not be achieved in the next decade. They might, in fact, stay out of reach. Okay, so I'd like to talk about a few of these today. One is that individuals from ancestrally diverse backgrounds will benefit equitably from advances in human genomics. So this continues to be an aspiration, but it's not where we are right now. Another is that research in human genomics will have moved beyond population descriptors based on historical social constructs, such as race. And lastly, again, there are 10 of these, I'm just sort of picking 3. An individual's complete genome sequence, along with informative annotations, will, if desired, be securely and readily accessible on their smartphone. So this is the future, in some sense, as the NIH kind of hopes it to be. And again, I'll use this as an organizing principle for this talk to tell you some vignettes from my group, as well as some ideas about where we can head in the future. And so I'll first start by focusing on these 2 ideas. So why is it that this is still something we continue to aspire to? Why have we not achieved it yet? So you may know, if you've looked at genome-wide association studies in, say, the EBI catalog, which Ewan talked about yesterday, that across over now 6,000 studies that have been deposited in the NHGRI EBI catalog, these test for correlations between mutations 1 by 1 genome-wide and complex traits or diseases, that over 90% of the samples represent only 16% of the actual human population and a subset of our genetic diversity. So this is, in fact, an old figure from work by Alicia Martin, who's now at the Broad Institute and has spoken out quite a bit in her career about this work. Of course, this has been something that's been discussed since about 2006, maybe a little earlier. But in fact, when a series of papers in around 2016 started to come out about this, the problem has actually, in the biobank era, gotten kind of relatively worse, right? So we have more and more data sets. They're getting bigger and bigger, but they're still not really going to rectify this relative imbalance. So in looking at this, one question that a population geneticist might ask is, how exactly can we use evolutionary processes like population histories and methods in order to identify disease and trait-associated mutations in a way that achieves that goal of equity, right? In making sure that research is really valuable to everyone. Another way of thinking about this that I was surprised to learn about when my group started looking at this a couple of years ago is that, in fact, more data actually leads to less analyses, in some sense, in medical genetics of diverse genomes. So even while the field and while many people who are here have been pushing along with funding agencies to diversify data sets, in fact, data from non-European ancestry individuals are really left on the table quite routinely. Right? And in fact, in the second half of this talk, I'm only going to talk about such data, but so this is, an issue for everybody. So there have been a lot of people who've been talking about this for a while. One is Simone Gravel at McGill, who wrote this really nice paper that was sort of based on an informal survey of UK biobank studies as well as studies of the health and retirement study in the United States that just noted how few studies, despite getting access to the full data, do any kind of analysis on the minority populations, and in fact do not cite any reason for not doing that. There's no requirement of mentioning even why this might be, and in contacting some of the authors, the reason they gave, which might make sense, especially to statisticians and computer scientists as well, we're underpowered, so we're just not going to analyze these groups. My group scraped PubMed for abstracts since the release of the UK Biobank in 2012 that mentioned the UK Biobank, and then we sort of incorporated a lot of our undergraduate researchers to check these papers by hand and tabulate how many of them between 2012 and 2020 actually reported any association outside the non-white British, outside the white British cohort. And based on our close reading, only 20% of UK Biobank studies report any association outside the white British cohort. This is usually in the South Asian cohorts, but there are tens of thousands of individuals who say have African ancestry, for example, in the UK Biobank that just tend to not be analyzed. And certainly analyzing the full data set is quite rare. Okay. So all of this to say that there's data being left on the table, and at the same time, GWAS itself, particularly marginal SNP associations with complex phenotypes, are really not giving us a lot of insight into complex trait architecture. The first time I think that this was identified really beautifully at scale was by Bogdan Pasanuk's group at UCLA. So this was based on heritability estimation, and heritability, that is Narrowsense heritability contributing SNPs, and where they lie across the genome with respect to quantitative traits, for example, in a complete blood count. So what you're going to see here is chromosomes on the x-axis, and the fraction that regions in those chromosomes contribute to the Narrowsense heritability of about 25 different complex traits. So these box plots are based on contributions to traits. And basically, you can see that these are sorted by contribution to estimated heritability, but they're essentially sorted, if you can see, I know it's hard to see in the back, by the size of the chromosome. So essentially, if the contribution to Narrowsense heritability is proportional to the size of the chromosome, except one anomaly is chromosome 6 that contains the major histocompatibility complex, which underwrites and influences many diseases. What this tells us is that, in some sense, associations that influence traits are distributed somewhat uniformly across the genome if we look at the SNP level. Right? And Jonathan Pritchard's group commented on this very soon after in their paper posing the omnigenic model for complex trait architecture. But they sort of mentioned one important sentence that kind of drove my research for the last few years and many collaborations, which is that one key question that's been understudied to date is the extent to which these influential variants, or causal variants we might call them, are spread widely across the genome, or are they clumped into disease-relevant pathways. Okay, so there's obviously been a lot of literature on this, on burden-based testing, for example, or SNP-level aggregation, and I want to tell you a little bit about what we've done in that space. And much of this is influenced by thinking about the statistical model that GWAS is based on. So if we think about the traditional GWAS framework as a classification problem, where I'm just going to use these words quite loosely, so forgive me a little bit about saying causal in kind of a non-causal way, but if we try to think about the goal of GWAS as differentiating causal SNPs from non-causal SNPs, then many statisticians, beginning with Bradley Efron in 2007, but Matthew Stevens particularly in 2017, have talked about the strength of this assumption and what it induces for us. So what it's actually saying is that we think that effect sizes are drawn from two different distributions. For the non-causal SNPs, they come from this point mass at 0, whereas for the causal SNPs, we think they come from some other Gaussian distribution, right, that is centered at 0 maybe, but has quite a large variance. And we can think about this as testing the hypothesis that the proportion of variance, phenotypic variance, that's explained by a particular SNP, we're testing the hypothesis that that's 0 against this alternative that it's something non-zero. But the truth is that this is a really specific null, right? And it means that with enough samples, The statistical signal that we can get, which I think is the world that we sort of live in now, is that almost everything is associated with a trait of interest, right? So how do we sort of sift through these results with ever-growing data sets? So inspired by some of Matthew's work, a recent student from my group who's since graduated looked at the UK Biobank and did a lot of simulations using real genotype data, but spiking in sort of causal associations and developing synthetic phenotypes that were quantitative under a variety of heritabilities. And just to illustrate this idea of what happens to false discovery rate in GWAS, you can see it clearly in simulations. So here I'm just showing you power of GWAS to identify these truly associated spiked in effects in simulation. So the power is quite high. Obviously it goes up with sample size and with Narrowsense heritability increasing, which are the two bars on the right? So you can achieve even something like 80% power, which is good. But at the same time, you're achieving a really high false discovery rate, right? Because GWAS is essentially calling everything as positive. Now, so again, this is an illustration of this issue of false discovery rate, and that's what's difficult to control in GWAS. And it gets even harder when you start to get good resolution on rare variants, for example, right? Because those can often look, look funny in the GWAS setting when they're combined with common variants. So along with a colleague, Lauren Crawford, in biostatistics, we developed a method we call Genie, where we sort of altered the null hypothesis a little bit. So now we're trying to just reduce the false discovery rate, keep our true positive rate, but reduce that false positive rate. And basically, we alter the null hypothesis a little bit, saying, suppose instead we assume that we're testing a null of beta being approximately 0. So we're going to allow for some noise around that delta mass. So let's suppose there's a third category that we're going to call interactive SNPs, and these co-vary with the causal SNPs. And they have effect sizes that might be indistinguishable from those that are emitted from causal SNPs. And we want to kind of control for these and then separate them from the targets that we focus on. Okay. So this is what this model looks like now, and sorry that it's a little bit cut off over here. But related work was done by Nilanjan Chatterjee's group at Johns Hopkins in 2018, where they considered exactly 3 components, so exactly this model that's being shown. What we do is have an arbitrary number of components that we can learn, and we basically just constrain them so that the putative causal classification component has the widest variance, right? And then we're constrained by that mass at 0. So another way of formulating this hypothesis is we're trying to learn some threshold sigma epsilon squared, and we want to classify everything coming from a component with a bigger variance than that as causal. So I'll just tell you a little bit about what we do in Genie kind of at a high level without going into the algorithm too much, but first we focus on some of what Matthew has done, which is shrinking inflated GWAS effect sizes using regularized regression. We use ElasticNet. We recalibrate this null hypothesis, which you can again think about as looking for this epsilon-based threshold. We then fit a K-component Gaussian mixture model with successively smaller variances to learn that threshold, and we've done that in different ways over the last few years. And the real insight in Genie is to aggregate the SNP level effect sizes into gene level effect sizes, or SNP set effect sizes. And actually, what's I think really nice about this paper is that these are written in such a way as to estimate the heritability that this set of SNPs actually contributes to the trait. So there's sort of an interpretable component to it. And it also uses the covariance structure of LD among the SNPs. The idea is to look at them kind of insist together as a null distribution for correlations in the effect sizes. So that's really what's new here. So other methods, we test against Genie in simulation, and again, using real genotype data from the UK Biobank. And just to show you a little bit, some of these might be some usual suspects that you've heard of. So using kind of burden tests or things like the SCAT family of tests and MAGMA, you can see that both Genie and then RSS, which is Matthew's group's method, outperform these other approaches. RSS, though, is really computationally intensive because it's fully Bayesian, which is obviously like the way one wants to go in this world, but our EM offers a kind of empirical based speed up here. And this is really zooming in these ROC curves on the false positive rate being quite small, a max here of like 0.1 Similarly, in terms of precision versus recall, we do quite well, even when there's subtle structure that's confounding the effect sizes. So you can run this on effect sizes alone, summary statistics, and even if those have a little bit of structure in them, Genie can, because of that null, using the correlation in the LD landscape, can control for that. So just going back to this idea of false discovery rate, what exactly is happening in these types of methods that try to aggregate beyond the SNP? So I'll just show you again what was happening with GWAS that we talked about earlier. Okay, Genie does really well. Of course, I'm going to show you a scenario where Genie does well. But I think what's interesting here is conceptually what these methods are doing that are moving beyond the SNP. So first, what's happening here with SCAT is that simply aggregating across a set of SNPs allows us to reduce our state space, our number of tests, and it lets us reduce our false discovery rate as a result because we're getting kind of additional lines of evidence of enrichment for association in a particular region. Then we get from RSS the benefit of shrinking the inflated GWAS effect sizes, okay, which is also very useful in controlling false discovery rate. And then LD allows us to more than have the false discovery rate on top of that. So understanding how we expect these effect sizes to be covarying with each other is, I think, a really useful conceptual addition to this repertoire. Okay, so part of why we developed this approach was to go back to this question of how do we think about multi-ancestry GWAS in an efficient way, right? Data is not solving the problem. So how do we think about methods as a way forward in order to analyze data sets that we have and truly analyze them fully? So, what we were inspired by was the idea that given that LD is differing across ancestries or other ways we can partition individuals, and I use partition lightly because obviously ancestry is not necessarily going to fully partition individuals, and individuals can have mixed ancestry. How do we conduct multi-ancestry GWAS studies? So I think what's interesting when we think about the GWAS model is that it's really focused on one particular, at a first approximation, and one particular ground truth. And I think now we're getting the data with which to interrogate this question. So it could turn out that I am wrong in questioning this as a ground truth, but I think it's important to realize this is what we're looking for in the discipline when we do GWAS, which is definitely a good place to start. It's that the true mutation level trait architecture is the same for all individuals, right? And we can think of many examples where this is largely the case, and there's certainly been a lot of work put into the fact that it seems like the same SNPs underwrite a lot of traits to a first approximation, at least a large number of them. But you could imagine a series of kind of nested models that allow for genetic heterogeneity, which has come up in this meeting already. So you could imagine that, for example, the same genes underwrite a trait, but with different causal mutations. And this is a biologically plausible model. Obviously, there are many traits that we can think of and are taught about or teach in our own classes that exhibit this, but certainly effect sizes do seem to vary in African ancestry individuals. For many traits compared to non-African ancestry individuals at a degree that GWAS does not uncover without sort of targeted testing in those groups. Similarly, you could imagine ancestry-specific modifiers of which core genes are perturbed, say they're all in the same pathway. So this seems to be true, for example, with how populations deal with hypoxic environments, that there are sort of different solutions in, say, Tibet versus the Andes. Skin pigmentation also seems to be one of these traits. And lastly, we could maybe just for completeness imagine, a totally different kind of scenario, although I would say, generally this last scenario is kind of there's less evidence of this in the literature. But again, maybe we have the data coming, especially whole genomes, high-quality whole genomes, that could let us ask this question. So this was the subject of a paper that came out recently from my group, was to kind of interrogate all of these models using these sorts of gene level or SNP set association tests. So this was led by a former student of mine who's now postdoc with Arbel Harpak at UT Austin, and gathering a lot of data together. The UK Biobank, Biobank Japan, and also PAGE Consortium study data was part of this work. And this was a really great effort on Sam's part, and also really exciting manuscript collaboration, particularly with the PAGE Consortium. So these were all quantitative traits, and then again, one thing that we've done in collaboration with Ben Raphael over the years is take these gene-level approaches and then try to identify gene subnetworks using graphs like protein-protein interaction networks to identify connected components that are enriched for associations. Here, taking Ben's work that's been really exciting in cancer and then applying it to common diseases. So just to show you what some of these results can look like and how they might look a little bit different from what we see in sort of traditional GWASs, I'll show you one example. So we find a large gene subnetwork of apolipoproteins that's enriched for associations with triglyceride levels in multiple cohorts in the UK Biobank and the PAGE Consortium. So here's what some of these graphs that are found to be significantly enriched for interacting genes that are themselves enriched for associations. So the colored in genes are ones where we find ancestry-based gene-level association test results that are significant, controlling for tests across all genes. And there are some overlapping sets here between, say, UK Biobank and Biobank Japan. But there are also some ancestry-specific signals that we find that are connected within this subnetwork that have been validated in other studies, usually smaller, more targeted GWAS studies. For example, this association with BASE1 that actually is associated with a comorbidity between triglycerides and Alzheimer's disease. Similarly, in the PAGE study, which is enriched for, or designed to study indigenous admixed individuals in the US, this very small group of Native Hawaiians also has some significant results with this pipeline, particularly associations in diabetes that, again, also are associated with a comorbidity in this group that's quite prevalent, even in this small sample, of high triglycerides along with diabetes. So this might be something one would see in a multi-ancestry GWAS anyway, but sort of as like separate lists of genes and maybe even these graphs presented separately. But we can also do the test now because in part of gaining power by reducing the number of tests at by looking at the gene level, we can also see that all of these signals are connected to each other kind of biologically. And so this is an example of what I think bringing this evolutionary lens to association studies can offer us, and also getting genetic heterogeneity modeled kind of as part of the pipeline really offers us a way ahead to think about both highlighting the shared architecture of traits across individuals, but also having some resolution to identify potential ancestry-specific alterations. So this is really important because prediction is obviously one of the projects of human genetics, and many of you probably know, but if you don't, I want to tell you that predictive power for complex traits is really low, right? And we might expect, especially computer scientists might expect that, learning out of sample is going to be hard when your discovery set is kind of representing one ancestry. Maybe we should assume this to be the case. But this is in fact true even within the discovery cohort ancestry. So even in the UK Biobank white British individuals, which are almost 400,000 people, the ability to predict within that trait within that ancestry for different traits is variable. And this has really been highlighted by Molly Chavorsky's group and also Jonathan Pritchard. So here are examples for instance of sex-specific alterations of particular traits, particularly cardiac traits, and also age-specific differences in prediction or even heritability estimation has been shown by Gil McVean's group for things like BMI. Right? And there have been a number of people who've been working in this area, particularly with respect to admixed individuals like Bittarello and Matheson. Harpak and Chavorsky have talked about how to model changing environments and change our models to realize that association, even with the same stable optimum for the phenotype, is going to differ when the environment is sort of shifting. And Sri Ramachandran's group and others like Noah Zeitlin, along with Richard Border, have talked about the importance of assortative mating and shaping these kinds of results. So I think it's really important to realize that those of us who are here in this room have the opportunity to contribute to this, because if we really want to improve trait prediction and also understand gene bioenvironment interactions, then we really need new methods and we need new models, and we're not going to get there with data alone. Okay, so some future directions in this area before I shift gears. The classification scheme that's in Genie, where we take SNPs and then place them in these various components, actually offers, I think, a way forward for unifying prediction and association. So that's something that we're working on now. We're not going to do as well as using the whole genome, but if we can interpret more of what we can predict, then I think that could be an interesting contribution to polygenic scores, and maybe there are others in the room who are interested in that sort of extension. We also are interested very much in multivariate GWAS and in heritability estimation and how to prioritize traits for phenotype prediction. So perhaps the traits we're trying to predict are not the right traits to try to predict. I think the Border paper does a really nice job of highlighting this, and my group has had a couple of forays into this, including a talk that was spotlighted at Recom last year. And I think after spending many years motivated by the idea of doing multi-ancestry GWAS, I actually think what I've come to realize, and maybe many of you have already realized, is that ancestry is in some ways it's only one way to think about partitioning human data sets, but it's perhaps the least interesting way. And so I think another way to think about this is to look at differences across, say, sexes or age cohorts or other aspects of proxies for environments that might influence complex traits. Okay, so now I want to totally shift gears, and I'm using, again, this strategic priority from NHGRI as a very loose organizer here. So this is largely unpublished work from my group, but the idea that's inspiring the next part of the talk is that many of us and many of our, say, friends and family members already have access to aspects of our genetic data through various direct-to-consumer testing products, and also, kind of an awareness of biobanks. And so what can that help us learn as methods developers about genomes? And this is basically focused on identity by descent, which we learned about yesterday a little bit in the genotype to phenotype session. So this is probably a review, but just to be on the same page, a way we can think about IBD, in this case, identity by descent, is as identifying regions of the genomes that are largely identical, tolerating some errors, that two individuals have inherited from a common ancestor. And these segments will be large, but they'll decay because of meiosis each successive generation. Okay. So we can imagine, thinking about calling these IBD segments, and IBD sharing between a pair of individuals is going to depend on that pair's genealogical relationship. So we think about the genome as being approximately 3,500 centimorgans, where a centimorgan is approximately 1 million base pairs. Then this is the picture of a painting of what IBD might look like between a grandparent and a grandchild. So they're going to share approximately 50% of their genetic material identical by descent, but it's going to be distributed throughout the genome in sort of different ways that are determined by recombination. Distant relatives, this decay happens really quickly, distant relatives are going to be unlikely to share IBD segments. So first cousins are, sort of close relatives. The reason why there are so many bars here is it goes like first cousin once removed, second cousin once removed, and so on. But if we're looking at individuals, the percent of pairs with this particular relationship that's highlighted who share at least one IBD segment that's more than 5 centimorgans long. So that's just one segment, 5 million base pairs, which is a pretty good chunk of DNA. You can see that first cousins will definitely share that amount. It goes slightly down as you get to third cousins. And once you get to eighth cousins, you'll have very few relatives who share such a long segment of DNA. However, we now have large genetic biobanks, and these are full of relatives, distant relatives. So not relatives that any of us would identify as relatives, but individuals with whom we have shared ancestry, going back about 8 generations. This is a plot generated by one of my students, Cole Williams, who I'll talk a lot about today, that's based on predictions that were written down by Schur and Nielsen in this very nice Journal of Mathematical Biology paper. So it's basically, based on biobanks and the proportion of the population that's being sampled, what proportion of those participants are going to have different degree cousins in the data set, with first degree cousins being the bottom line, that's red, and 8th degree cousins being the top, that's purple. So if you think about the goal of many biobanks, particularly in Europe, is to sample a non-trivial percentage of the population, 1%, 5%, right? So now there are going to be a lot of these relatives. So what's happening in biobanks is distant relatives are more common, and even small data sets will have very many distant relatives. This is true in the 1,000 genomes, for example. And close relatives will be less common than distant relatives, but increase in number as we increase sample size. Okay, so as an aside, my group has worked on IBD abnormalities for a little while, in particular in a collaboration with 23andMe that was published in 2019. So maybe just because some of you have done these kinds of products, you could, I'll show you a little bit about this particular project. So one thing that a direct-to-consumer company like 23andMe does is if you share your profile with an individual, but particularly if you tag an individual as, say, a parent, then you have reported back to you genetic segments that are IBD with that individual. Right? And this is based on an algorithm that Brenna Henn developed and wrote down in 2012. So here's an example that inspired this project of a customer's product that was released to them and posted on Reddit. So that's why I'm posting it here. This is just part of the IBD results between this individual and their father. Okay, so here you're going to see a few chromosomes, and you see that chromosomes 11 to 15, what's being reported to this individual is they are half identical to their father, which makes sense. They're supposed to share 50% of their genetics IBD with their father. But on chromosome 16, there's sort of a strange thing that's reported, and in fact what's being reported is on just that chromosome, this individual shares no genetic material IBD with their father. So one of my students sort of saw this and was an intern at 23andMe in the time, and then we started this project. And in fact, this happens kind of non-trivially in the data set, as I'll show you in a minute. So this is a signal of something called uniparental dysmia, which is the inheritance of one chromosome fully with no IBD representation of another parent. This happens due to correcting an error in meiosis that can happen early on in the formation of gametes. So what's really interesting about this from a cellular biology standpoint is that it highlights that there are ways that, for example, a trisomic gamete can be corrected to promote proper disjunction in development. And this had never been studied in a non-clinical setting. So everything that was known about uniparental dysmia was based on individuals who presented in the clinic with imprinting disorders, usually. So because 23andMe is so large, there are about, at this time, now it's grown, almost 1 million duos, which includes trios in the data set. And we found 199 individuals with uniparental dysmia, again, just doing this data-driven approach of trying to look for an absence of IBD sharing on a single chromosome, and in some cases, even multiple chromosomes. So we report for the first time the per chromosome rates of UPD in a sort of general sample that is a non-clinical sample. Obviously, this isn't a random sample. And because of using duo information, we're able, in fact, to assign this to different sides of the family, paternal versus maternal. Enrichment here is on chromosomes that are known to have imprinting disorders. At the same time, these individuals aren't reporting, at least in survey data, any enrichment for those kinds of disorders. So we estimate for the first time the overall rate of UPD as actually 1 in 2,000, which is not as rare as I might have thought it to be. But the existing estimates, which were from clinical cases, were quite different. So this is based on a data set that's maintained online, started by Lear, and these estimates range from about 1 in 3,500 to 1 in 5,000. Maternal UPD is 3 times more common than paternal UPD, and I don't have a good explanation for that, but again, because it has to do with this kind of ability to correct for a meiotic error, but it's an interesting feature of the data. And we observe enrichment. And the last thing I want to say about this kind of side project is that we also look at the UK Biobank. We also find putative cases there. Again, we develop a classifier that I'm not talking about in both 23andMe and the UK Biobank to look at individuals without parental data. But I think what's really interesting here is that thinking about the rare genetic disorders that we can learn about and that data sets like GNOMAD and EXAC have taught us about, we actually can learn a lot about the strength of embryonic selection. So the fact that these are live-born individuals already who are adults, the median age of customers in a data set like 23andMe tends to be 50 years old, already says that they've kind of reached a point where embryonic selection didn't operate in their development, and so somehow they're not representing both parents on a chromosome, but it's not leading to significantly deleterious consequences. Okay, so now let's just talk about IBD sharing in the UK Biobank more broadly. So again, I'm only going to focus on the white British individuals here, but importantly, I developed in part to represent about 1% of the total white British population, at least this part of the sample. So the average white British individual in the UK Biobank shares at least 5 centimorgans with about almost 25,000 other individuals in the data set, and there's a pretty large standard deviation on that. So some individuals share that much IBD with even more individuals. If we instead threshold to something like second cousins, if we look at total IBD sharing of about 35 centimorgans, they share that with still about almost 24 other individuals, again, with a very high standard deviation. So there are some individuals who share this much DNA with 100 individuals, for example, in the data set or more. But on average, about 95% of these relatives only share one single long IBD segment with, say, a focal individual that we might focus on. So our goal in this unpublished work is to classify these IBD segments that an individual shares with their relatives, and we want to classify them as coming from either the maternal side or the paternal side. Or I'll frame this as thinking about two sides of the family, side 1 and side 2. If we have sex chromosomes and kind of enough metadata, maybe we can classify these properly as maternal versus paternal, but just imagine we're thinking about two sides. So why might we be interested in inferring the parent of origin of IBD segments? I think there are a lot of really interesting applications of this. The first is to improve long-range haplotype phasing. So this can really help if we can stitch across IBD segments. We could identify switch errors, for example. But we can also enable inter-chromosomal phasing quite easily if we have close enough relatives who share IBD across chromosomes. So that's the application we're focusing on now. But there are many others. One is parent of origin effect GWAS studies. So if we want to combine, say, transmission disequilibrium tests along with sort of large sets of distantly or unrelated individuals, IBD could give us a way forward. And lastly, we could do things like understand parent-specific local ancestry inference. So again, sorry I'm talking so much about 23andMe, but just because my own results are uninteresting as a South Asian person. Here's an example, again, of the kind of thing that the product might share, which is where certain local ancestry tracts come from when they entered a person's ancestral genealogy. So now imagine that instead of just knowing, okay, at this time, British and Irish ancestry entered this 23andMe employee's own personal history, suppose one could say this came from the maternal side of the family. That might be sort of interesting to study at scale. Okay, so now I'm going to tell you a lot about what Cole's been thinking about, which is an algorithm for bipartite clustering of IBD segments. So the idea here, which has been used by some other phasing algorithms recently, but I'll tell you about what makes ours different, is to represent these IBD segments as a node in a graph. And let's just assume that we have really good intrachromosomal phasing, although we can kind of stress test that a little bit. But the reason why I'm going to assume that is, again, we're looking at these data sets that have like millions of individuals in them, and just phasing intrachromosomally is really high quality in that scenario. So now you could think about this graph as having positively weighted edges between IBD segments that were inherited from the same parent, and negatively weighted edges. So this is one new thing we're drawing on, which is the negatively weighted edges. So then our goal is to do bipartite clustering, and we're going to use the sign Laplacian matrix of this graph. And this is an approximation to the NP-hard problem of the minimum ratio cut on this signed graph. So here's a point where I would love to get some feedback, because this was one of the methods that we found that had come up with a heuristic for doing this. But I'm sure this group knows about other ones. So this was a recent paper from a SIEM data mining proceedings by Kunegas et al. But the idea here was actually inspired or illustrated by some sociological data on relationships between tribes in Papua New Guinea. So here the positively weighted edges had to do with friendly interactions, and the warring tribes have negatively weighted edges. And again, actually in anthropological studies and sociology literature, we find a lot of statements of this problem, but not actually approaches for clustering on it. So our goal, again, is to use IBD sharing to pull apart these two sides of the family using genetic segments. Okay, so let's just say we start with good statistical phasing on a couple of chromosomes, but we could even maybe tolerate some switch errors, right? So now imagine that we call IBD on, sorry, this is slowing down so much. Okay, so now we're going to call IBD segments that are more than 5 centimorgans. We could actually reliably do more than 2.5 centimorgans, but let's just say we're going to do 5 for a focal individual with other individuals in the UK biobank. So we're looking at the focal individual's chromosomes, and now we're assigning IBD to all these other relatives, A through J, that are in this data set. Okay, so those are indicated here. They have varying lengths, but they're sort of approximately co-located in ways that are determined by recombination and the strength of the relationship. So now we could take these segments and use them to form tiles that indicate overlapping sets of IBD segments, and we have to have kind of a threshold of how we stitch these together, right? But then what we'll do is we'll take segments that are in the same tile, but on different haplotypes, and place them on different subtiles. So this is where having the phasing is important within the chromosome, but again, this can allow us to understand phase across chromosomes, which is a kind of nice thing that this does. So we're going to assume that these IBD segments in different subtiles are inherited from different parents within the same chromosome. Okay, so now we can label these based on how many IBD segments are being shared with a particular relative. So this is kind of a merging of the segment with the individual that's in the data set. So now our goal is to create an IBD graph. So now we're going to add edges between nodes that are in the same subtile. So they're in the same tile and also on the same haplotype. And then we can add negative edges between nodes that are in different subtiles on the same tiles. And then the last step is to add positively weighted edges between nodes that are corresponding to the same relative. And we can weight these by overall total IBD sharing, which can range a lot in these data sets. So now we can do some Bipartite clustering. And actually what's really nice about this is it gives us pedigree collapse as part of the approach. So here you can see that there is a green edge connecting two individual segments from one relative that are placed on opposite sides of the graph. And that's because this is an individual who's actually shares ancestry IBD with this focal individual from both sides of the family. This is something we're quite interested in from a population genetic perspective. So we're starting to validate this in both the UK Biobank and also a broader collaboration, but just to show you some results from the UK Biobank, we use a test set of 1,050 individuals who have both parents in the data set. So again, another thing that's interesting to think about with these large biobanks is the fact that there are individuals who are parents and children in them, even with the kind of design of the UK Biobank and its enrollment. So these let us validate the method. So here what's happening is there's quite a bit of variability in how much IBD individuals share with each other. But when we add all the edges, we cluster a lot of IBD segments. And in fact, we can cover almost 90% of a lot of individuals' genomes just with these kind of very distant and a few, actually not even that close relatives, right? So with largely eighth cousins, there are some individuals for whom they get incredible coverage of the genome, and you can kind of recapitulate it without parental data. If we employ an edge inclusion heuristic particularly focused on total IBD sharing, say to second cousins, then we really improve our precision. So here this red dot is showing this median value. So again, we can like cover reliably the genome, although maybe less of it. So we cluster fewer IBD segments overall. Okay. So there are lots of, I think, future directions to go in this area, but through the population genetic lens, obviously one is to apply the methods to just individuals without post-validation, without using any, we don't use parental data, we're just leaving it out in order to determine ground truth, as well as to look at diverse ancestry. So how does our coverage vary? Does it increase in some groups that have more LD, sort of as in the background or not? Obviously, inter-chromosomal phasing can be possible here, which is really nice. And a really exciting new collaboration we're developing with Simone Gravel's group at McGill, and I know Shadi Zabad is here. He didn't talk about this work, but he's done some amazing work on simulating ancestral genealogies through a structured diploid pedigree. And I think understanding these evolutionary processes that lead to pedigree collapse and how that might vary across ancestries particularly, but in the context of the Quebecois genealogies that Simone's group has done a lot of work on, is really interesting. So the last thought to leave you with is that LD is everywhere. It's really affecting a lot of things in human genetics that we don't talk a lot about openly, I think, as a field. Another example is tests for selection. So this is a common set of results that will be reported in sort of traditional outlier scans for selection. And the cross-ancestry results and the fact that there are more putative targets of selection that are identified in groups with more LD is something that I think more people should be pushing on. Of course, groups like Adam Siepels and individuals, in Jerome Keller, especially like thinking about some tree-based data structures, are also thinking about this question. But I think this is an interesting other one that I'm not talking about today. So I'd like to end there and just thank my wonderful group that I'm very privileged to work with. I told you a lot about Cole Williams today and also about Wei Cheng and Sam Smith. And I'd be happy to take any questions. Thanks so much for your attention.